sort of as a representative of the MoTrPAC Bioinformatics Center, I'm here today to talk to you about some of the data that is now publicly available from MoTrPAC. So we've looked at the multi-omic response to exercise training across rat tissues, and this data is now available through the MoTrPAC Data Hub. So first, just some brief background. Physical inactivity is, of course, considered a major cause of many chronic diseases, while regular exercise is known to reduce risk of premature mortality, improve overall health, metabolic health, and also reduce the risk of the same chronic diseases. And exercise has shown to have a positive effect also on the treatment of at least 26 different diseases. It's also examples of such diseases are, for example, Parkinson's, type 2 diabetes, hypertension, COPD. So there are lots of different effects of exercise, and clearly whole body effects, as we do see effects from everything from the brain to the heart to the muscle. But there is currently a limited understanding of the underlying molecular mechanisms by which exercise promotes health and prevents disease. And this was one of the sort of key aspects that the NIH Common Fund had in mind when they designed the MoTrPAC study. So current exercise literature when it comes to more large-scale omic studies are limited. We can see a few of the biggest studies to date here. We have at the top here the classic heritage family studies that was conducted in the 90s. So here they had a big sample size, so over 600 individuals, and they looked at muscle for a subset as well as blood, and it's focused a lot on genomics, so we can see the different omes studied here. So for heritage genomics, there's been a targeted metabolomics assay as well as a plasma proteomics study that was published quite recently in Nature Metabolism. We have a couple of examples here from looking at acute exercise in blood with multiple omes, but only 36 people. One study from Robinson that looked at different modalities of exercise, because that's also a lot of variability in what type of exercise people are looking at. So this study looked at 12 weeks of training in 72 individuals, had one tissue, so skeletal muscle, and looked at multiple omes across this one tissue. And then, of course, we also have animal studies. So this is one acute endurance study that looked at 8 tissues, which is way more than most other studies, and looked at metabolomics assays. So let's get into what MoTrPAC is and what we hope to contribute in this field. So MoTrPAC is a multi-center NIH Common Fund project. It started in the end of 2016, and the sites are distributed across all of the US. It's divided into a preclinical phase that focuses on rats. So primarily rat studies, and you can see some of the preclinical animal study sites that have conducted those studies in green there. There's also then a clinical phase with human data. So the red here are the clinical sites that are recruiting the human subjects, and there's also a pediatric site for pediatric participants. And then there are a lot of different chemical analysis sites that run the many different assays that we do in MoTrPAC to investigate all of these different omes. There is one bioinformatics center that I represent here at Stanford, and we sort of gather all the data from both the clinical and preclinical sites as well as the chemical analysis site to process all of this data, work on analysis in collaboration with the entire consortium, and also share this data through the MoTrPAC Data Hub. And there is also a coordinating center and a central biorepository. So there are tissues available also for people who are interested in utilizing these studies down the line, even if you're not a part of MoTrPAC. So to the study design a bit. So if we start by looking at the human study, the main human study consists of sedentary adults that do a lot of different phenotypic assessments. They are divided into 3 different groups. So there is a sedentary endurance intervention, a strength intervention, as well as a non-trained control group. And what these people do is they undergo 1 acute exercise bout of either endurance or strength, or then no exercise if they're in the control group. We collect muscle, blood, and fat, so 3 different tissues from the humans. And then they undergo 12 weeks of their respective training modality, and then repeat these phenotypic assessment as well as the acute bout with biospecimen. So they perform this acute bout of exercise both in their untrained state and then after 12 weeks in the trained state. There is also a pediatric cohort, which is similar to the sedentary. Main difference is that they only provide blood samples, so no muscle or fat biopsies, and they only do endurance training and not resistance. And then there is also a highly active cohort. So the goal is to recruit about 300 highly active participants that are either resistance or endurance trained, and then do 1 bout of exercise with muscle, fat, and blood sampling. The sedentary and all these clinical MoTrPAC arms, so recruitment is still ongoing. We had a long break due to COVID that delayed things a bit, but the goal is to recruit about 1,800 adult individuals. And then we're in the process of analyzing the first batch of data right now. But today, I will mostly focus on the preclinical animal data. So the reason for that is that this data is publicly available already. So you can go and look at it right now and download and do what you want with it. So for the preclinical data, or at least part of this is available, the full preclinical animal study in MoTrPAC has 2 different age groups. So 6-month-old adult rats and 18-month-old older rats. These do either a single bout of acute exercise or a long-term training program. So the long-term training program that I'm going to talk about today for the 6-month-old male and female rats, they have trained 5 days a week at 70% of their VO2 max for 1, 2, 4, or 8 weeks. So there's also a time series component to the training. And from the animals, we collect a lot more tissues compared to the humans. So that's also one of the strengths with the animal study sites. And if you're interested in learning more about the study design for MoTrPAC, there was a descriptive paper published back in 2020. So going back to the exercise omics studies, what's MoTrPAC hoping to contribute? So if we start with the rat study, we have way more tissues than any previous study. So 19 different tissues. And all cover all of these different omes except genomics. The human study, when they are coming, the goal is to reach somewhere around 2,000 people in total, 3 different tissues, and then again covers all of the omes. So this is what I'll focus on today. And if we just look a little bit more closely at the study design for this. So this is the animal endurance training study in MoTrPAC is a progressive running protocol over 8 weeks. So these are now for the male and female 6-month-old animals. So we have the same also for the 18-month-old. And then there is a sedentary control group that remained untrained for 8 weeks. And the tissues were collected 48 hours after the last exercise bout, and then sent away to these chemical analysis sites to be run for different omics. And this rat just displays these 19 different tissues that were collected and the omes that were assayed for each tissue. So not all tissues have all of the omes. But all of them have at least 2 ohms. So transcriptomics and metabolomics has been run for all of them. So to some of the results, if we start by just looking at some of the phenotypic changes in these rats, the VO2 max test, here I just show for the control group 4-week and the 8 weeks training groups with males in blue and females in pink. We can see that both males and females increased quite substantially in VO2 max with the 8 weeks of training. So significant in the 8-week groups. As expected, there was also a small decrease in the female untrained group over the course of these 8 weeks. For the body weight, there was no change in body weight for the males. However, the females did increase over the intervention in all 3 groups. We also did some body composition analyses. So here you can see the percent body fat. So the males, the trained males, decreased in their body fat percentage, whereas there was no change in the females. However, the untrained females did increase in body fat percentage. So in the females, training kind of rescued this increase in weight. So they remained weight stable over the 8 weeks. And here you can see the lean mass. There was no significant difference in lean mass in the males, a small decrease in the untrained females. So moving into the multi-omics, we conducted 2 main differential analyses. One looking sort of overall at the training-induced changes, basically asking the question, is an analyte changing in either sex at any training time point over the course of these 8 weeks of training? And second, we looked at the time-wise changes, where each then training time point is compared to the sedentary control, separated them by tissues and sex, to see if there are time point specific changes across the training. So we start by looking at the training regulated analytes overall, and this shows for all of the different tissues here. And now I'm just showing the transcriptomics data, which we have for all. You can see we've separated blood and plasma. So transcriptomics was run for blood. And the numbers that you have in here correspond to the number of differential training regulated transcript in that particular tissue. The color corresponds to the proportion of differential to analyzed transcripts in this case. So we can see the greatest transcriptional response was seen in the adrenal gland and the colon, which was somewhat unexpected. But we see changes across pretty much all tissues. And if we expand this to all ohms, the white squares here represent, it shows that there is no data for that particular time point. So at the top here, we see the 2 epigenomic assays. So we've looked at the methylome, DNA methylation modifications, attack sequencing for chromatin accessibility. And this was run on 8 different tissues. You can see here, transcriptomics that we just saw, and then we have the protein and protein modification assay. So we have run proteomics on 7 different tissues, phosphoproteomics also on 7 different tissues. And then there are 2 tissues that have also been analyzed for the acetylome as well as the ubiquitinome. So heart and liver are the 2 tissues that have been assayed the most across all the different ohms. We also looked at a targeted immune panel, as well as several different metabolomics assay, a mix of targeted and untargeted assays. So in total, we found over 35,000 analytes that were regulated with training across the assay tissues and ohms. So if we move over to the time-wise changes then, in order to try and visualize this in a more interpretable way, we performed a clustering analysis where we simplified the differences and by classifying them into 9 possible states. So basically, an analyte could be, differentially regulated or not in males or females or both, and it could be up or down regulated. So the 9 possible states then is, for example, an analyte can be upregulated in females only in either of these 4 different time points that we have. They can be upregulated in both males and females, upregulated in males alone. We have the corresponding downregulated features here, no change. And then, of course, an analyte could also be oppositely regulated with upregulation in females, down in males, or vice versa. So yeah, you can see the meaning of these here. So if you go to look into the MoTrPAC data, you will see a lot of these types of plots to be able to identify sort of the main time trajectories in the data. So as an example, if we're looking at an analyte like this, that is not changing in females up till 4 weeks, and then it increases in females after 8 weeks. And then the same analyte then increases in males already after 1 week, and then it keeps being upregulated across all the training time points. This analyte's trajectory would then translate to an increase in male only after 1, 2, and 4 weeks of training, but then be upregulated in both males and females after 8 weeks. So some questions that can be asked using these graphical clustering data is, for example, what analytes increase in abundance across both the sexes at 8 weeks? So you can hone in on the 8-week time point and look at this particular node and see, okay, what analytes are here? What are the pathway enrichments for that? You can look at what proteins that decrease across all time points in females alone. You can look at the top trajectory for a certain ohm in a certain tissue. So this is just an example of the gastrocnemius muscle results. Here, just showing 7 of the states, so not the oppositely regulated, simply because there are very few features that are oppositely regulated in the gastroc with training. So this shows the top 5 trajectories across training. And the edges here are colored based on the ohm. And the larger or the wider the line, the more analytes belong to that line. So we can see that the biggest trajectory in muscle consists of analytes that increase already after 1 week of training, and then they remain upregulated across all time points and in both males and females. So mostly sex-consistent results. And again, these nodes also are size specific. So a bigger node corresponds to more analytes. So we can see the green one is the biggest line here, which corresponds to proteomic changes. And then we also see a downregulated cluster here, or trajectory in both males and females, where there are a lot of phosphosites associated with this. So the orange line corresponds to phosphoproteomic changes. So I thought I'd just go through some of the findings that we have from this very large resource. So there obviously you can dig deep into whatever tissue or sex or ohm that you're interested in. But I'm just going to give a few examples of physiological changes and that we could identify from the multi-omic data. So we start with looking at some of the whole body responses to training. So we were interested to see, are there omic changes that occur across multiple tissues? And this is a pretty busy slide, but this focuses on the 6 tissues that had the deepest molecular profiling. So white adipose, liver, skeletal muscle, heart, lung, and kidney that had the most assays. This just shows for the transcriptomic data, but you can look at it for all the omes. And what we saw was that the majority, so this is a classic set plot. So the largest number of differential genes here was seen in the white adipose out of these tissues, and they were unique to white adipose. So the majority of the differential genes were specific to each tissue. But if we're looking then at the overlaps to see, okay, what are some of the common responses across tissue? We have one here comparing that is differential in both lung and white adipose tissue. And you can see some of the enrichments here. So they are mostly the common responses between lung and white adipose are associated with cell adhesion and immune responses. We have some that are common here between heart and skeletal muscle, which perhaps expected. And here it's a lot of pathways related to mitochondrial functions. Looking at differential transcripts that were regulated across all of these tissues, there were only 22 genes that changed across all of these tissues. And these were associated mostly with the heat shock response. So the heat shock response was regulated mostly, and also mostly actually at the protein level. So obviously, as I mentioned before, improved metabolic health is a critical adaptation to exercise. So we also wanted to specifically look into the metabolic adaptation across these tissues. So this figure shows the metabolite enrichment results from the metabolomics data. Just looking at the 8-week response, we see, for example, that liver had the largest number of enriched classes of metabolites, a lot of different lipid classes. Also down here, you see, for example, pyrimidines. One example from this enrichment is a 1-methylnicotinamide, which is known to play a role in inflammation, for example. We saw that lipids were also affected in the heart and skeletal muscle. So these blue ones are the lipid and lipid-related compounds. And so here you have the skeletal muscle, for example, which has a lot of different lipid classes that are enriched, and the heart here. So this is trying to look overall at some of the main sort of metabolite classes that were affected with the 8 weeks of training. We also wanted to look at the effect on metabolic proteins. So this figure shows a gene set enrichment analysis that focuses only on mitochondrial pathways. And we see positive enrichment for multiple mitochondrial pathways, especially in these metabolically active tissues like the heart, skeletal muscle, white adipose, liver. So a lot of red for upregulation of these, or enrichment of these, positive enrichment of these pathways. And we see this primarily for the proteome. We also looked at the acetylome for heart and liver, where we have that assay available. And we see a lot of mitochondrial proteins that are increase in acetylation with training in these two tissues. So it's clearly regulated both as the protein abundance as well as the acetylation level. When we compare what proteins these are, because obviously if you get an increase in the protein abundance, you can then expect to also get a similar increase in acetylation, but it turns out that these are actually largely different proteins. So it's not the same proteins that increase in acetylation as increase in protein abundance. So focusing in on the liver a little bit, this shows the largest trajectory in the liver. where we have an increase is specifically in males up until 4 weeks, and then after 8 weeks in both males and females. So these plots have all of the individual analytes that follow this trajectory in them. And if we look at then what pathways that are represented, or what pathways that are enriched for the analytes that follow these trajectories, we see that this is largely associated with amino acid metabolism. And here the purple circles, each circle is one pathway. So this just shows the overall classification. And purple means it's an enrichment at the protein acetylome level. We can see that mitochondrial metabolism is also mostly affected at the acetylome level, but also at protein abundance. We also have peroxisome and lipid metabolism that's affected. So we do see quite a substantial regulation of lipid metabolism in the liver. And we see it both when we look at protein abundance and protein modification data, as well as lipid changes from the lipidomics data. So you can sort of try and get across all of these different ohms to look into the potential changes in tissue function with training. For all these analyses, we can also compare the responses in males and females. And we did observe substantial sex differences in multiple different tissues. So this is, again, a pretty busy slide, but one of the key differences we saw between the sexes was a difference in the immune pathway enrichment. So we saw sex-specific responses. So if we look here, these are different immune-specific pathways. The triangles here, if there is a gray triangle, it means that there is a sex-consistent response. If it's a pink triangle, it means it's a female-specific, and blue means it's a male-specific enrichment. And then the triangle can point down for down regulation or up for up regulation. So what we can see here is, for example, in the white adipose, we see a lot of pathways that are affected exclusively in males. Also here, where you see an opposite regulation in males and females. Same in brown adipose, where we do see an upregulation of a lot of different immune pathways that is specific to males. We also saw in the small intestine that showed downregulation of a lot of pathways that was primarily specific to the female animals, so female rats. And so a lot of different immune regulation differences between the sexes. You can see some of the individual trajectories here for brown adipose, as well as white adipose between males and females. We also looked at the different cell types to do a correlation. So this plot to the right here is based on a correlation between markers of immune cell types, and then the features that you can see here from a transcriptomic perspective. And what we see is that there is a very like a male specific increase in immune infiltration in both brown adipose and white adipose. So there seems to be a sex specific shift in the immune cell types, which is something that would be interesting to look further into. Another key sex difference that we saw was in the lung phosphoproteomic data. So looking at lung phosphocytes, we can do a similar to a gene set enrichment analysis you can do with post-translational modification. So this is a post-translational modification set enrichment analysis, where we look at the different phosphorylation sites that changed and map that back to known kinases. So kinases that are known to affect some of these sites. And we did find, so this just compares the normalized enrichment score for some of this PTM-SEA results. And we can see, especially in the 1 and 2 weeks or early post-exercise or post-training time points, there was a big sex difference particularly in PRK8CA, which is the catalytic subunit of the protein kinase A. So it's involved in a lot of different signaling pathways. But this was down regulated in males, but upregulated in females. So we can see the trajectory here. We see this early upregulation of associated phosphocytes in the females, and the same time point, these phosphocytes decrease in males. And down at the bottom here, you can see this the individual some of the individual phosphocytes that are driving this different in enrichment. So a lot of these proteins are involved in cellular structures, so like cytoskeleton, cell-cell junctions. So this suggests a sex-dependent role of protein kinase A in mediating changes in lung structure or mechanical function with training. So if you're interested in this data, so I just showed a very small part of all of the results that we have from these rats across tissues. All of this data is already available through the MoTrPAC Data Hub. So you can go in, don't need any registration or anything. You can just go to the Data Hub and look through download data that you're interested in, look through different visualizations, search for your favorite genes. So I will just show a little bit of a demonstration of some of the things that you can use it for. So these are recorded, but you can also just go in and look for yourself. So this is what it looks like when you come to the MoTrPAC Data Hub. You can read a little bit about MoTrPAC, and then up in the corner, there is a login option, but you don't need to log in to be able to explore data. So if we start by just looking at downloads, if you're interested in downloading the MoTrPAC data to analyze yourself, you can see here that there are, for example, already pre-bundled data sets. So you can download like all the phenotype data, all of the transcriptomics data, and just immediately click to download there. You can see that. And then, yeah, you see it starts downloading. So then you can get all the transcriptomics data and start looking at that. If you're interested in particular tissue or particular ohm, you can also directly get all of that. So here I'm just selecting heart and the 2 different metabolomics, targeted and untargeted. And then you can just say, okay, I want the merged tables, and just download those. And what you do then is you put in your email address and name, and then you will get an email notification when the files are just ready for download. So here you can just select whichever files that you're interested in, or whichever data. If you don't want to analyze it yourself, you can also explore and go to the gene-centric view. Here you can search for 1 or 2 or many genes. Here, just as an example, citrate synthase. You can then narrow this down to whatever tissues you're interested, like gastroc and heart, kidney. If there is a particular ohm, so like RNA-seq, global proteomics, for example, you get those, and then you can immediately compare the results for these tissues that you've selected. So here, for example, selecting the proteomics data, you can then see the time-wise trajectories, what happens in males and females with citrate synthase protein up there in the kidney, and then down there in the gastroc, you see a significant increase in both males and females. And then you also have the heart down there. So whatever tissues you're interested in comparing, you can just, or whatever ohm, you can immediately get to that data. And yeah, and you can just update and see, okay, I'm interested in seeing if there were any acetylation sites that changed on the citrate synthase protein, and just click there, whichever sites you're interested in, and again, compare directly what happens with these different lysine acetylation sites. Okay, so next, I'm just going to switch view. You can also directly interact with the graphical clustering results. So if you go to explore again, and you go to graphical clustering, there is both an option for the landscape, which includes everything, but also a mitochondria specific, if you're interested in looking at the mitochondrial changes. And here you can just select whichever tissue you're interested in. Again, we can take the heart, and hopefully you recognize these plots now, where you have all of the different trajectories. You can also see the number of features and also the ohm distribution of these biggest trajectories. Here you see the top 5 in the heart. You scroll down, so you can see the biggest one is upregulated in both males and females. You get them split by ohm. So these are the sequencing based. Here you see the proteomics with abundance, phospho, and ubiquitinomics, and metabolomics. And here you can again zoom in on, for example, this largest trajectory and look at some of the pathway enrichment results that are also divided per ohm. So for example, here we see enrichment for both acetylome, phosphoproteome, as well as protein abundance for TCA cycle. We can see the individual trajectories. And then here you see the downregulated, mostly affected at the transcriptome. So you can just scroll through here and see. So you have all the data for these top 5 trajectories, as well as for the top 5 nodes. So this is the biggest node in the heart, all the analytes that increase in both males and females after 8 weeks. So you can see the enrichments for those, pathway enrichment results. See the cellular response to heat stress that we also saw before when looking at the results. TCA cycle, again, is affected across multiple ohms. So you can do a lot here, depending on what you're interested in. We also have an interactive data visualization. So this allows a bit more freedom to look at what you want and also to compare different tissues using these cluster visualizations. So here you can select whichever tissues you want. I'm just sticking to heart and gastro for simplicity. You can select ohms. So yeah, whichever ohms you're interested in. So I'm just doing the phospho, the global proteomics. And then you can immediately go in and see those clusters. You can decide how many paths you want. We have 13 paths. If you just want to see the top 5, you can do that. So this is the top 5. And now again, it's for gastroc and heart. And it shows, so you get a distribution also between the tissues as well as by ohm. Maybe you recognize these. So the feature trajectories, you could have to select the cluster. So now I just selected the 8 weeks for males and females. So you can see each individual, one line is an individual analyte. And I think this is the coolest feature with this part of the tool, because here you can directly interact with the pathway enrichment results. So if again, select the 8-week males and females, so all the analytes that increase after 8 weeks in both males and females, and again, now it's both heart and gastroc. You can then zoom in and you can look at the pathway enrichment, the TCA cycle, and then immediately see which analytes that are driving this enrichment. So both proteins and transcripts in both heart and muscle. You can see here many different enrichments, carbon metabolism, glycosylate dicarboxylate metabolism, and you can just play around with this and immediately compare then what's affected in the heart and what's affected in the muscle at these different omics levels. You can also pull those out and move them around and play with this however you want. So mitophagy only affected at the transcript level in the heart, for example. If you only want to see what's common between tissues, you can select that multi-tissue pathway only and see those that are affected for both heart and skeletal muscle. And again, you see like PPAR signaling is affected at the protein abundance level in both heart and skeletal muscle. So you can see if there are any common proteins that are regulated. Here we have the heat shock proteins again. So a lot of different pathways, and immediately get access to which are the drivers behind those. And you can also see the different time-wise trajectories. If you're specifically interested in seeing like, okay, what changes over the course of the training for a certain trajectory. And again, you can just deselect and compare whatever tissues you're interested in. Okay, so to summarize, this is an unparalleled multi-omic resource for exercise adaptation across tissues. So we have 19 different tissues, 9 ohms. It's over 90 data sets, and we find over 35,000 analytes that change with training in at least one tissue in ohm over the training type course. So we really show that this is a organism-wide effect of endurance training in these rats. You can, of course, interact with the time-wise data using these graphical cluster visualizations, so you can compare time-wise trajectories across tissues, or if you're interested in only the early 1-week time point or only the 8-week time point, you can zoom into that and you look at only those analytes. I only showed a very small selection of the main biological results, but we do see responses in most organs. We see substantial sex differences, particularly in the white adipose, the adrenal gland, the lung, and the small intestine. We see a lot of metabolic adaptations across multiple tissues. So mitochondrial pathways were affected in many different tissues. And importantly, we also provide a lot of associated tools to explore and interact with this data. I mostly showed the MoTrPAC Data Hub today, where you can do everything from downloading the data yourself to searching and visualization of your favorite analytes or look through visual clustering results. But I also want to highlight that there is an R package available, or 2 R packages actually, if you are interested in analyzing the data in the same way that was done for the paper that is coming out fairly soon. You can do that. You can also utilize that for creating the visualizations and doing the analysis for your own data. So with that, this is, of course, a huge consortium. Of course, want to thank the NIH Common Fund for providing the funding and the infrastructure for us to be able to do this. There are tons of people involved in this consortium, and can't thank every one of them. But I'm simply one representative of this group, and the work that I presented here today is the work of many excellent scientists across these different institutions. So thank you very much for coming, and be happy to take questions.
Okay, great. Thank you for Dr. Lindholm's presentation. So now it's our Q&A session. You can unmute yourself if you have any question, or you can continue to use chat function. So I just see there is one question from Dr. Swapandas. So what was the rationale for choosing rat as the animal model in this case? Only one inbreed strain of rat was used, which neglects role of genetic architecture on exercise response. How the rat data is going to be useful for genetically devised human?
Yeah, that's an excellent question. So the main reason for choosing rat is tissue amount compared to mice. That of course another option for a small animal. To be able to run all of these omes, especially on tissues that are limited in size and therefore amount, rat was the sort of predominant choice over mice. And also because this is a rat strain that has been shown to result in robust changes in VO2 max with training. So that was sort of the physiological rationale. And this rat strain was one that could be provided by the National Institute of Aging. So the NIA colony has this strain. And could provide the enough rats that we needed for these studies. So those were some of the aspects that we considered. The genetic diversity is clearly not huge in this rat strain, and we also only have omics data from 5 to 6 animals per time point, sex, and ohm. So it is limited from that perspective. There are, of course, efforts like, for example, Andrea Heavener from UCLA, who's looking at many different strains of mice and comparing the genetic influence of training adaptation. So that's another question. That's something that has to be addressed with future efforts. But to be able to cover all of these tissues and all of these ohm from a cost perspective, it was not possible to also do multiple strains, unfortunately. And of course, also 2 ages. So we also have the 18-month animals that are coming.
Okay, thank you. Does that answer your question? Yes, and there is another question from chat for direct message. So is this portal accept other research to deposit their data, or they have to be a consortium member?
So for now, you have to be a consortium member, but this is something that is being discussed. So it's something that might be open up as a possibility in the future, but that's something that we need to, discuss with the NIH if that's going to happen or not. But we have already included actually some other related data types from other studies.
Thank you. Yeah, you can unmute yourself or use the raise hand function if you have any question, or send me the message, then I will see, will ask question. Yeah, and then there is another question from the chat. Are there any tutorial available to show us, like walk us through the portal, how to find like top signal pathways, for example, like the immunology pathway that you mentioned during the talk, that I could watch or show my students?
Yeah, that's also a great question. And yes, there is a tutorial. So far, we have a very basic tutorial on how to navigate the data hub. So that's already available. And we are working to implement more specific tutorials as well to sort of exemplify some of the different features depending on what you're interested in. So more tutorials are coming as well in association with the publication, hopefully, of these papers soon.
So are those tutorial right now posted on the portal or through other channel?
Yeah, so there is one tutorial at least already available on the portal. And then we have one also for the R package that is, I believe, not available yet, but it's coming very soon.
Okay, and there's another question from Swapan. So how do you plan to use this multi-omics data for causality analysis?
Sorry, for what type of analyses?
Causality, I think. It's in the chat, causality.
Oh, causality analysis. Oh, okay. Yeah, that's a great question. I think obviously, I mean, we are limited by the descriptive nature of this. And a lot of to firmly establish causality, I think we'll definitely need to do follow-up studies with more targeted questions and perhaps do cell modeling, etc. We've done a little bit of that already, but this is really to be seen as a data resource where you can test. You can use it for hypothesis generation. So if you have a gene or protein of interest and you want to see what happens with exercise at different levels of regulation across these ohms, you can use that, and then from there to firmly establish causality, you'd have to do more targeted studies. And one of the resources available for that is also the biorepository, so the MoTrPAC biorepository with tissues. But yeah, that's definitely a limitation.
Okay, I hope that answer the question from the audience. And there is one comment from Dr. Shah. It's just telling everybody there's also open hours if people are interested in learning more on data or portal function. And all the information are on the MoTrPAC Data Hub website.
Yeah.
And then also there's one question from chat just coming now. So you show us the enrichment pathway during the demo. Is there any download function if I want to work offline for the result?
Yes, you can download enrichment results as well. They are mostly available right now as supplementary data to the paper, I believe. I don't think you can specifically download that from the data hub yet. So those will the enrichment results will be coming with the paper. But yeah, I can look into that if there's a way to actually get it right now from the visualization tools. I'm not exactly sure.